He has testified at hearings in the U.S. Senate and Congress, the U.S. EPA, the California State Assembly, and in the European Union. He also served on the National Academy of Sciences Committee on Hormonally Active Agents in the Environment. He is a fellow of the American Association of Advancement of Science. His research, which is funded by the NIH and private foundations, concerns the long-term consequences for the brain and reproductive organs of exposure during fetal life to normal, natural hormones and both man-made and naturally occurring endocrine-disrupting chemicals. Please join me in giving a warm IAOMT welcome to Dr. Frederick von Saal.
What I'm going to do today is tell you about something that a number of years ago in 1996 really became an extremely controversial topic. Then at the time, a person, Nicolia, a Spanish scientist, chemist, published a paper in Environmental Health Perspectives saying that dental sealants were able to leach out into saliva at reasonably high levels, and the levels generated a lot of controversy. And the American Dental Association did a follow-up study to this that Dr. Olia had a lot of problems with because his was done in situ in looking at dental sealant application in a person's mouth. And the follow-up study was done in vitro. And of course, anybody who's ever used these products knows that the ability to create a very high level of polymerization in vitro is different than what you're going to achieve in a person's mouth. And the Jada refused to publish the Olia commentary on this, which he then was forced to publish in another journal, Environmental Health Perspectives. In 1999, he finally published a response to this paper that was published by a person named Nathanson. So all of this conflict was going on, and actually I'm going to take a somewhat different tact, and instead of talking about specific products and their rates of leaching, what I want to do is just talk to you about what we've learned over the last decade about the health effects of the chemical bisphenol A that is a fundamental part of dental sealants that are used to protect teeth. And one of the important aspects of this chemical is it's been known since 1936 that it had the efficacy of estradiol, the natural endogenous estrogen. And it has been classified as an endocrine disrupting agent. A group of us got together in 1991 and came up with this term. And it's an exogenous agent that interferes with any aspect of endocrine function, whether it's production, release, transport, metabolism, binding. So it isn't just a matter of hormone mimicking chemicals. There are many different ways to disrupt endocrine function. And there's two major issues here. One, the regulation of homeostasis in an adult, and developmental disruption. And my focus really is on developmental disruption because if this estrogenic mimicking chemical were a really serious problem for adults, then women would not be able to take birth control pills and go off of them without severe problems. So while these chemicals are in the body of an adult, they do disrupt physiology. I mean, there's no argument about that, just like a birth control pill does. And make no mistake, bisphenol A does everything a birth control pill, estrogen, would do. But when its exposure occurs in an embryo, developing infant, a child, anytime prior to the achievement of full development, then there is the possibility of permanent disorganization of that developmental process. And so I'm going to be talking about a couple of chemicals, bisphenol A, estradiol, and the drug diethylstilbestrol that was administered for about 25 years to pregnant women on the mistaken belief that it would prevent miscarriage. It turns out it didn't do that, but it caused cancers, reproductive abnormalities, immune dysfunction, just wreaked havoc with the offspring, and it didn't do anything to the mothers of a permanent nature. These things are characterized by hydroxyl groups and at the end here that allow them to slide into the estrogen receptor binding pocket and activate estrogen responses. The potency with which these chemicals activate responses are a little different. Estradiol and bisphenol A are fairly equally potent. For some responses, bisphenol A is a little less potent. Other responses, it's equally potent. The reason that you people need to be concerned about this is that in all sealants made with either dimethacrylate, diglycidyl ether, bis-GMA, or all of these products have various components where there are ester bonds present. And where there is an ester bond, esterases act on them, and they're subject to temperature, increased hydrolysis, and particularly as you move off of pH neutral, either towards acidic or basic condition, then you accelerate hydrolysis. This will result in the release of free bisphenol A. And this is, again, a estrogen mimicking chemical. The question is, of course, and this follows up somewhat from the last talk, because I face some of the same issues, is does Paracelsus rule and only very high toxic levels matter, or do low levels make a difference? What we are in, and you just heard a little bit about what is called a paradigm shift in science. We are in the state of a paradigm shift in the field of toxicology. It's very easy for me because I'm not a toxicologist. I'm a developmental biologist who studies the role of hormones in development. But basically, the tradition of toxicology is to look at acute toxicity in an adult of very high doses. However, the endocrine system can never be studied that way, and I'll tell you a little bit about that. And now we're thinking about, my God, if there are chemicals in the environment that act like sex hormones, we can't study them the way toxicologists study acute toxic chemicals. It's a totally different approach has to be taken. And that's because if you look at the hormone estradiol, you see something really quite stunning. First of all, from an acute toxic point of view, when you get up into the microgram per milliliter, part per million range, which of course a millionth of something seems like a very small number, but that's an acutely toxic range of this chemical, estradiol, natural hormone, in human breast cancer cells. But at a million-fold lower dose, below a part per trillion, in the part per quadrillion range, this shows that as you add this to human breast cancer cells that have estrogen receptor alpha in it, and I'll come back to that, one of the particular types of receptor, from multiple types that are available, then below a trillionth of a gram per mil of medium, you can stimulate proliferation of breast cancer. Okay? That's not good. So this gives you an idea of the phenomenal potency of the base hormone that we're going to use as the reference for looking at the potency of bisphenol A. So when somebody says to you, bisphenol A is really weak, it's not nearly as potent as estradiol, they're talking about relative to the potency of Superman over here. Because this is one of the most potent hormonal molecules known to science. All right? So if something's a little weaker than that, it can still be phenomenally potent. And that's what we're going to see is, in fact, the case. The other thing regarding the concept of Paracelsus is that acute toxicity predicts low-dose hormonal activity. And you'll notice in the last talk, he said that if you identify the acutely toxic dose and divide by 100, you're in a safe zone. Oops. It's only off by 10,000 to 100,000 when you do that to a hormone. High-dose testing has no relevance whatsoever for low-dose effects of hormones mediated through receptors, period. So we're in a scientific revolution that's going on. And one of the things that we're beginning to see is, with regard to this group, are the safety of products? Do they really need to be reevaluated? As we were seeing in the last talk, are there trends in human disease that would make us think that chemicals such as estrogen are becoming a problem that we're seeing showing up in terms of some various health trends? Well, these are some data published a number of years ago that really only go through the 90s. And some of these are from the dreaded CDC, but they do actually put out some meaningful studies. Others are from private research. This is from Shauna Swan. Some of these are from data collected from various countries. But what you see is testicular cancer, breast cancer, hypospadias, where the penis no longer has the urethra coming out at the normal tip, but it can come out at various parts of the penis, including the base of the penis in its most severe form. And sperm count, evidence for sperm count decline. One of the important elements here is that in all of these cases, there's evidence that estrogenic chemicals and other endocrine disrupting chemicals can actually cause this in animal and now have been implicated in published peer-reviewed studies to these trends in humans. One of the conceptual shifts is away from the tradition of toxicology of just looking at adults and looking at developmental effects. And the maxim of pediatric medicine is that children are not little adults, and that what goes on in an adult body cannot be used to predict what will happen when your child is exposed to a drug or a chemical. That's a no-brainer. Anybody who's a parent knows that. One of the important things that I'd like to mention is that for many years, people were focusing on mutagenic effects, thinking genes control development. Well, we have had a dramatic shift in the field of developmental biology. We're not looking at mutagenesis anymore in a classic sense. We're looking at what are known as epigenetic mechanisms. These are entirely different processes that actually determine what we become. Yes, genes are important, but after the Human Genome Project, since we don't differ by anything more than 2% from chimpanzees, people started to think, my God, what's going on? Okay, how come we're so different? Well, it isn't necessarily the genes you have, it's how they're turned on and off and how they function. And that's where hormones and these mechanisms come in. And for those not familiar with this concept of epigenetic modification, instead of thinking about substituting bases here in classical mutational events caused by chemicals, what we're seeing now is you take a transcriptionally active gene and its protein code around it, and now events such as hormones, bisphenol A, estradiol, and other regulatory signaling systems during development add methyl groups to cytosine bases. And in the process of doing that, if you deacetylate the histone proteins that surround this DNA, what you end up with is a transcriptionally silenced gene. That's how you start off with a single fertilized cell and have muscle, nerve, bone, all these different types of cells in your body with identical genes. Some genes have been shut off, some have been turned on, and it's like you have a rheostat. It's not just a total on-off event. The activity level of the gene is set through these differential epigenetic covalent modifications of genes that until we developed recently new analytical techniques, you couldn't see these things. By classical mutagenesis analysis, you wouldn't see any of these chemical modifications. So now what happens, whereas normally an estrogen receptor sits as a transcriptional activating protein associated with sequences of DNA, and you can activate gene transcription when an estrogen binds to its receptor, if you have methylation occurring in various regions of the promoter region of a gene, that gene transcriptional activity is blocked. Polymerase can't go down that, and you have the absence of transcription. And this is the kind of normal event that is occurring during development. And what we now know, this is kind of a busy slide, but it's important for the other things I'm going to be telling you. One of the things that people thought originally was that bisphenol A was a relatively weak estrogen relative to estradiol. And some of the cells that were looked at had the alpha form of the estrogen receptor. And we now know that if a cell only has this receptor expressed associated with its DNA, bisphenol A has a relatively low affinity for it. And of course, it also depends on the kind of co-regulatory proteins that are associated with the receptor, and it's the entire complex that matters. There are other cells in the body, such as the testes, sperm, the prostate, various regions of the brain, that express the second form, the beta form of the estrogen receptor. And then if other proteins that are called co-regulatory proteins are expressed, you can have the consequence of this be an increase by hundreds of fold in the potency of bisphenol A relative to what you'd see at this receptor. And then lo and behold, a couple of years ago, people finally realized that instead of just working in the nucleus by diffusing in here and binding to these nuclear receptors, there are uncharacterized receptors associated with a cell membrane that activate calcium influx and very rapid systems that lead to changes in transcriptional activity, but in a much more rapid fashion. And these are phenomenally amplified systems. You can have 1 million or plus fold amplification. So phenomenally low concentrations can lead to changes in gene activity. And in these receptor systems, bisphenol A and estradiol are absolutely equally potent. And again, during adulthood, these activational events of genes, the genes are activated, and when the hormone goes away, no more activation occurs. What is going on in the developing embryo is that not only are you activating genes, but you are permanently imprinting them through these epigenetic mechanisms. But these occur during periods of development prior to the time that organogenesis is finally complete, when your adult body plan is completely formed. So we have different explanations now for why bisphenol A was originally thought to be a very weak molecule, and now we're beginning to get evidence that, in fact, its potency is absolutely phenomenal. So how potent is it? Here's a recent publication where people injected into the cerebellum of a 10-day-old rat and doses of estradiol and bisphenol A, and you'll notice that just like human breast cancer cells, they were able to get effects of bisphenol A and estradiol that were absolutely equal at doses below a part per trillion. Okay, so this is very interesting. Another thing that you see happening here, the response occurs and then it goes away and then it comes back. And what is actually going on here on a mechanistic level is the lowest dose effects operate through opening up of calcium channels. And what is very clearly taught, I mean, I teach this in my introductory endocrinology course, is as the level of ligand, these hormones, increases by about 2 to 3 orders of magnitude, you shut down the capacity of the cells to produce the receptors, and the response to the hormone disappears. That's called receptor down regulation. We understand the molecular mechanisms of this. It's not hand waving. And then the second set of responses come back through different activational systems because this system is inhibited. So these, what are known as non-monotonic dose response curves, create real problems for classic toxicology because Paracelsus, The whole concept of the dose makes the poison cannot be applied to this hormone, to any hormone. There is no exception to that. I know of no hormone that doesn't produce non-monotonic dose response curves. But importantly, bisphenol A through these cell surface receptor systems is very potent. Now here's a situation from data from our own publications out of my colleague Wade Welshon's lab, where in human breast cancer cells, we initially found, as many people did, that there was about a 10,000 to 100,000 time difference in the potency of bisphenol A in human breast cancer cells, but they only express this nuclear ER alpha receptor, where we now know that for cells that express those receptors, they give us the weakest response of bisphenol A, but that's not characteristic of what happens throughout the body. And I just thought I'd show you some recent data from our lab. What we do is we go into a mouse fetus and we dissect out the fetal mouse prostate, and then these cells, called the mesenchymal cells, that actually control the development of prostate gland, they're the ones that have ER alpha, again, not ER beta. And what you see in the mouse, just like you do in the human, is this dose right here, which is expressed in molarity, is 0.28 picograms per mil of estradiol, and it's inducing the development of prostate, the gene that is making prostate androgen receptors, the receptors that allow the prostate to respond to testosterone. And one of the things we have shown is that this is through an epigenetic mechanism, that it imprints that gene to produce about 6 to 7 times the amount of androgen receptor in the prostate throughout the remainder of life of that mouse. And these mice end up with hyperplastic, benign prostatic hyperplasia and prostate cancer in adulthood. And we also see a positive feedback thing where at a slightly higher dose, estradiol initiates the induction of its own receptor. Well, bisphenol A does exactly the same thing, and it does it at about 1,000 times higher dose. But one of the interesting things about this, and again, this is in mouse prostate cells, and it does this at exactly in the range of bisphenol A found in human blood. And it also induces its own estrogen receptor. And what is interesting here is that, again, bisphenol A permanently increases by about 6 to 7 times the permanent increase in prostate androgen receptors. And if you take a hormonally responsive organ and you increase the sensitivity to testosterone in it, it actually, again, is a risk factor for cancer. And lo and behold, they end up with cancer in adulthood. One of the things we've now seen in a whole bunch of studies is there are about 20 studies showing that the range of bisphenol A in human blood and in human urine and human tissues is in the range of about 1/10 of a part per billion up to about 10 parts per billion. There's remarkable consistency about this throughout studies around the world. Right? It's really amazing. They're all in exactly the same range. And we are seeing that cells from animals and from humans are able to actually respond to bisphenol A at lower levels than the average person is exposed to. And even the lowest level found in humans is above the range that's capable of causing damage or cellular changes in animals. One of the things, of course, is to ask the question, how do you study long latency effects? You do something in a child, and then that child shows abnormalities later on in life. This is a very difficult challenge, and if anybody is familiar at all with the DES tragedy of the millions of women who were administered this drug, and it took 25 years of administering this to millions of women before one astute physician finally realized that he had seen in a couple-month period of time 6 teenage girls with vaginal cancer. and that had never been seen in a teenager before. And it took him months of time to finally realize all their mothers had taken this drug, and then they banned it. But that was after 25 years of it being a medically prescribed drug. So it's very difficult to link things that go on much later in life, like adult cancers, to what happened to a child. Come back to the link, does what goes on in an animal with DES, is that relevant to what happened in a human? This is a study that was published where the prostate gland was taken out of an early developing mouse and put with just testosterone and then a little bit of DES. And the glands, these chambers here, are the glands developing. So you're getting hyperplastic glands. So you end up with an enlarged prostate. And again, this treatment ends up with these animals having prostate cancer later on in life. And notice the dose of DES, less than 1/1,000,000th of a gram. 1/2 of a trillionth of a gram is able to do this in organ culture. 100 times higher level of bisphenol A, 50 parts per trillion, can do the same thing in primary culture. So that is a phenomenal potency. Yes, it's less potent than DES, but it is still staggeringly potent. What is important about that combination of information there is that there is virtually 100% concordance between the human health effects of DES to the millions of people that were exposed to it due to their mothers being given this drug during pregnancy. And when you expose mice to DES and look at the consequences in adult mice, there's virtually identical outcomes. So this is a matter of connecting the dots. DES and bisphenol A cause the same effects in mice. There are probably a dozen papers showing that. And DES causes the same effects in humans and mice. So we really have a reason to think that the animal literature is relevant to what we're seeing with regard to estrogens impacting developmental processes in children, infants, and fetuses in both animals and humans. So here you have data that show that the human levels of bisphenol A are in the low part per billion levels. And in all studies, males have higher levels than females because the glucuronidation, the conjugating and clearance process of this, there are feedback systems where there's a reduction in this in males due to higher levels of testosterone, which suppress this process relative to what goes on in females. And it's very easy to convince people that 1/1000th of something is a very small dose and not to be worried about. But remember, these are hormones that operate below a part per trillion. That's 10,000 times lower than this level. So while 1/1000th of something to most people seems like a small number, to me as an estrogen biologist, that to me is an astoundingly high number that in these other experiments is already into the inhibitory zone where you're suppressing receptor systems and inhibiting responses. And to me, this is a very high dose of an estrogen. One of the things that I'd like to point out is one of the reasons we started to study bisphenol A is my graduate student, Susan Nagel, was working as part of her PhD thesis on how plasma binding proteins block natural estrogens and regulate them from getting into cells. And much to our astonishment, we found that the high affinity binding protein, that's a major reason why estrogen levels are regulated in the blood and not all estrogen, naturally produced estrogen, gets into cells. Bisphenol A has virtually no binding to this protein whatsoever. Whatever you have in blood, the great majority of it can pass into cells, whereas the great majority of your naturally produced estradiol in your body is prevented from entering cells. So the potency of bisphenol A appeared to us to be much higher. And then we found out in the years since that in addition to binding to these nuclear estrogen receptors and having estrogenic effects, we now know that bisphenol A can bind to androgen receptors, and so can bis-DMA bind to androgen receptors and block the ability of androgens to function normally. This is essentially like giving a man with prostate cancer flutamide, the anti-cancer drug that blocks his prostate from responding to testosterone. Well, that's fine if you have prostate cancer. It's not so great if you're a pregnant woman and you're carrying a little male baby, because it's not going to come out looking like a male. Or if you have a child that's developing and you want that child to have normal levels of testosterone, and all of a sudden you're putting an estrogen and an anti-androgen in them, that's not so good. That's pretty easy to see. And then at even higher doses, it has anti-thyroid effect. So it has the ability to interact with many systems. And again, during the time of the fetus or the newborn or in children, when genetic programming is occurring, you not only regulate these genes, but you permanently imprint those genes through these epigenetic mechanisms and alter the functioning of this cell for the rest of the individual's life. One of the big questions was, are these dental sealants leaching? Well, the Centers for Disease Control, together with the US military, did a study on this. And this is JASCOW, it was published in JADA. And so just to give you, they took 15 volunteers from the military and who were going to have these dental sealants put on. And they were using two different types of dental sealant. And there's something very obvious from this. And that is, there is a background level that is present in urine or blood or tissues in the low part per billion range